right, let's keep talking about the cell cycle. And in this section, we're going to talk specifically about the S phase and how the cell gets ready for mitosis. Remember, we're still in interphase. All of these phases are part of interphase. All right, so the cell needs to make sure that after mitosis, when two cells divide to create daughter cells, each one has all the DNA it needs. So the cell is going to take the double strand of DNA, pull those two strands apart, and then complementary base pair new nucleotides to each strand so that it creates two double strands of DNA. These are called daughter DNA molecules. And the way it happens is there are specific binding sites called origin sites on the DNA double helix. Imagine these two lines of the double helix. And special molecules called DNA Polymerase bind to that, open up the two strands, and start to complementary base pair new nucleotides to the old strands. And then as they work their way along, eventually they do touch each other and then they release and they bind somewhere else and keep going until we have two complete daughter DNA molecules. I want to talk about this molecule for a minute, DNA polymerase. There are several different kinds of DNA polymerases. Don't worry about that. We're only going to worry about this sort of group of enzymes. It is an enzyme. Remember, ASE means that it's an enzyme. And remember that a lot of enzymes are named for what they do. So this one is no exception. DNA polymerase is going to make a polymer of DNA. And you remember that DNA is a polymer. It's a big, long molecule made up of monomers, and the monomers are nucleotides. So DNA polymerase connects nucleotides to create a strand of DNA. And in this case, it's going to connect those nucleotides to an existing strand of DNA to create a new double helix. And it actually does it on both sides. So we get two new strands. Okay, so that's happening during S phase, the synthesis phase, when we synthesize new DNA. All right, so during G1, before S phase, the chromosome was a single strand of DNA with this little centromere part here. After S phase, all of that DNA has been duplicated. We now have two double helices bound together at this little indentation called the centromere. These two strands are called sister chromatids, and when we finish mitosis, they will be separated, and each one is going to go into a new cell so that each cell has a full chromosome, a whole double helix, just like the cell that we started with. Okay, so we have the two sister chromatids. As long as they're held together at that centromere, they are considered one chromosome. Once they separate, then they become two daughter chromosomes. So we did G1, the cell was doing its thing. S phase, duplicate the DNA, end up with sister chromatids connected at the centromere. And now we enter G2. And G2, it's called a gap phase. And these G1 and G2 are called gap phases because the scientists couldn't see what was happening and they just thought the cells were like doing nothing. They're doing a bunch. They don't laze around. They got stuff to do. The most important thing that's happening during G2 is that the cell is proofreading the DNA. There's another enzyme that comes along, checks the original strand of DNA, makes sure that the correct nucleotide is complementary base paired to each nucleotide on the original strand of DNA, and fixes any errors. This makes sure that the DNA is correctly copied. Now, even with that, mistakes do happen, and we'll talk later about what some of those mistakes can do. Those mistakes can create mutations that can be passed down in generations if it happens in the right cells. So we'll talk more about that, but most of the time, the proofreading catches any errors. And once all that proofreading is done, the cell is ready for mitosis. The other thing that is happening during G2 is that all of the other organelles in the cell are being copied, because we're going to take this one cell, divide it into two cells. Each of those cells is going to need a full set of smooth endoplasmic reticulum and Golgi and lysosomes and all the other things. And so the original cell that's getting ready to do mitosis, it's going to duplicate all of that stuff in the cytoplasm in addition to duplicating the DNA. All right, so this is the end of interphase. Interphase, please note, is not part of mitosis. Mitosis comes next. The chromosomes are still just kind of lying around like spaghetti. They have not condensed yet. They're going to do that in the first part of mitosis. As mitosis starts, the nuclear envelope, still here, that's going to go away during mitosis. The centrosomes are these little T-shaped structures. They are duplicated at the end of G2 to make sure that we have two centrosomes. These are going to be vital to mitosis because they're going to make protein strands that are going to move the chromosomes around during mitosis. All of that is happening during interphase, and we are getting ready for mitosis. Now, here's one chromosome all condensed, ready for mitosis to happen, and it says to the other chromosome that's not condensed, dude, mitosis starts in 5 minutes. I can't believe you're not condensed yet. So get ready in this next section. Get all condensed. We're going to get ready for mitosis.